Welcome to The Mind, the Cure Protocol for August 11, 2026. No newly verified patient reaching milestone in this pass has reached care, so we will not pretend otherwise. We are following what changed in the evidence, what new research it opens, and what still needs proof before it can help anyone. This protocol is a living loop. People bring the questions that matter. Direct evidence changes the map. The source ledger remembers what we have already seen and checks the original date before calling it new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. Researchers reporting in Nature Communications introduced stPainter, a conditional generative model pre-trained on a pan-cancer scRNA-seq atlas that can enhance sparse spatial transcriptomics data to single-cell resolution without matched references or retraining. The broader significance is that this shifts spatial disease profiling from a data-limited measurement problem to an inference problem. AI becomes a general-purpose decoder that extracts cell-level state from incomplete tissue reads, an intelligence inversion that moves biology toward a phase transition where disease can be treated more like an engineering system than an observational mystery. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. A multi-center, randomized, double-blind phase 3 trial in Nature Medicine reported that Obinutuzumab beta, MIL62, a glyco-engineered type 2 anti-CD20 antibody, reduced disease activity in aquaporins for IgG-positive neuromyelitis optica spectrum disorder, a severe autoimmune demyelinating disease, compared with placebo. The result matters because it shows how a programmable immune-targeting intervention can convert a historically refractory neurologic disease into a tractable engineering problem, an example of the broader phase transition from empiric treatment to mechanism-led disease control, where AI-enabled biology can accelerate the discovery, validation, and optimization of curative strategies. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. Aureka Biotechnologies announced a $100 million Series B, bringing its total funding to nearly $200 million to build a biological world model for drug discovery. The signal here is not a single therapeutic result but an intelligence inversion. Instead of discovering drugs one assay at a time, the company is trying to make biology legible to machine reasoning, which is the kind of phase transition that could turn disease from a stochastic search problem into an engineering problem. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the Cure protocol stays current without pretending the work is finished.